The goal of this procedure is to temporarily and remotely silence neuronal activity in discrete brain regions while animals are engaged in learning and memory tasks. This is accomplished by first loading the AAV that contains a designer receptor and a reporter into a syringe for infusion. The second step is to infuse AAV into the region of interest in the rat brain. Next, the biologically inert designer drug clozapine N-oxide is prepared and injected systemically into the rat. The final step is to conduct behavioral testing using a sensory preconditioning paradigm. Ultimately, immunofluorescence microscopy on the rat brain tissue is used to verify placement of the designer receptor.
This method can help answer key questions in the field of behavioral neuroscience, including but not limited to regulation of motivation, attention, and learning and memory.
Ensure that all institutional precautions for working with adeno-associated viruses are followed throughout. The virus used in this video is classified as a Biosafety Level 1 compound. Prepare a 10% bleach solution for decontaminating AAV waste. Place a beaker containing bleach solution and a sterile 10 microliter syringe in the workspace. Place the microcentrifuge tube containing an aliquot of AAV into a standard bench vise. Load a 10 microliter syringe with at least 4 microliters of AAV. Ensure that there are no air bubbles in the 4 microliters of AAV solution in the syringe.